People are streaming in the door. Okay. Hello, friends. It is my absolute pleasure to welcome everybody to our Thursday seminar. We're here on March 25th. And it's my absolute pleasure actually to be welcoming somebody who's like not part of our department, but is part of our Georgetown universe. And if there's a point in time in which we're all back again in Regents, I believe that she's going to be a neighbor. What floor are you on, Esther? Fifth floor. Okay, fifth floor biology, folks. So Esther's your neighbor up there. Esther Braselmann's just joined us here at Georgetown University. She came to us from doing a postdoc out at UC Boulder. Before that, she'd earned her PhD at the University of Indiana. No, sorry, the University of Notre Dame. Sorry, at Notre Dame in Indiana. But as she's originally from Germany, where she did her undergraduate work. She's a biochemist, and she's a biochemist like in a really pure sense, meaning that I think if we didn't know better, we'd really think she was a biologist because she studies RNA dynamics, but she brings to bear on that study some really amazing chemical and microscopy tools. And so I got to know her a little bit last summer because one of the first things Esther did upon joining us at Georgetown was join us for our journal club last summer that we were holding for undergraduates, getting them to think about kind of cutting edge research and also to think about DEI issues in the sciences. And so I found myself in a room with Esther one day in that journal club. And it was delightful to get to know her, to know of her passion, not just for science, but for students and education. So now I've kind of talked on too long. I'll say she's going to tell us, I think, a fascinating story today about this particular platform called Riboglow for looking at RNA dynamics. I've read some of her papers now, and I'm fascinated by it. At heart, she also is a little bit of a microbiologist. So selfishly, I'm thrilled she's here. Sean's really excited because when I say microbiologist, I don't mean yeast people, your sort of microbiology or my sort with Giardia, but she studies bacteria. So I'm going to monitor the chat for Esther, and I know she's got kind of a couple of chapters of her story. And I think she and I talked, and she's happy to take questions whenever, but also I think that some of those break points between chapters of the story will definitely take a pause and gather questions. So Esther, welcome to the biology Zoom department at Georgetown University. It's delightful to have you.
Thank you for having me. I'm really excited to share my work with you all. So like Heidi mentioned, I'm really a biochemist by training. So what I'm interested in thinking about is how biochemistry is implemented in living cells. So if you think about live cells, we can think about a microscopy image of cells like this here. But if we think about a movie, the cells, this is going to work. The cells are actually going to move around hold on. This is supposed to be a movie. The cells are actually going to move around and be quite heterogeneous, which means that if we think about biochemistry from a textbook perspective, all the reactions that we know from textbooks will happen in these cells at the same time. But clearly they're implemented quite differently and quite heterogeneously in the cell. Okay, the cell, what does the chat say? Okay. I'm sorry, this computer is, can you still hear me? My computer is being really slow with this.
Yeah, we can hear you, Esther.
Okay. So this is sort of what my training has been like all around since I started grad school, where I was working on protein folding, but we were interested in how protein folding is implemented in living cells. So what we think about as protein folding from a textbook perspective actually happens in live cells. Similarly, I'm now interested in thinking about RNA dynamics. So there's lots of textbook knowledge about RNAs, but how does this actually happen in cells? And then what I did, so I think I'm going to, my presentation is crashing. I'm going to restart. Can you see all of this?
We can see it. We can see it crashing here, Esther. We can take a moment for you to restart it.
Okay, I'm sorry about that.
No, I think in 2021 technology issues, I believe possibly your computer recognizes that spring break starts in a day and a half too. It is like just a little on early vacation here.
Let's see. I think my slide deck is just getting too too large. That is what is happening. Let me try this again.
I think Esther, probably just speaks to the incredible beauty of your images, that you've got too many gorgeous images built in.
It's the movie that is the problem. Let's see. Okay, this is the movie I was talking about.
Perfect.
They're moving around. So then the question is, how do biochemical reactions lead to cellular function? And again, like this is where my training comes in. I'm trained as a protein folding person, but now I'm interested in RNA. And again, we can think about this from a textbook perspective, but all of these things happen in the live cell. So what this means for my postdoctoral studies is that I develop tools to visualize and track these processes in living cells. And that's what the majority of, so that's what I'm going to be talking about today, what I did in my postdoc. Like Heidi mentioned, my lab is being set up right now. So this is sort of my past work that will lead into my future work. So when we think about labeling biomolecules, we could think about organic synthesis. So these are small organic molecules that have different fluorescence properties. And you can see that organic chemists have been quite great at synthesizing them and inducing different features that might be useful, like changing colors, reducing cell toxicity, inducing cell permeability, etc., and those types of really practical applications. But nature has found a way to do this already. So fluorescent proteins have been discovered. So this is this barrel here is green, is GFP, green fluorescent protein, which folds. And if you look into the barrel, you can see the organic structure that we've seen on the previous slide for organic synthetic compounds as well. So now what we can do is tag our favorite protein of interest with GFP or another fluorescent protein and see where it goes within the cell and look at its function. A big caveat though is, which will be important later, that if we want to tag and label and track our favorite protein of interest in the cell, we have to make sure that by tagging the protein itself, we don't change its functional dynamics. So with that, so this is what Heidi mentioned, like there are breakpoints after each topic here that you're welcome to interrupt me. I want to talk about both my biological model system and then introduce a really short story about tagging and tracking secreted proteins. And lastly, I want to talk about my work on developing fluorescent RNA tags. So the biological model system I'm interested in is bacterial infections, which I found is really exciting because it really features all the things that I think are important to look at. So it's a really complex and heterogeneous system. I'm specifically interested in Listeria monocytogenes. It's a gram-positive bacterium that's the third leading cause of death from food poisoning. And you might have heard about Listeria from different food recalls because it can be found as a food contaminant quite regularly. So you might have heard about news like these. So why is Listeria dangerous? It turns out that Listeria can survive in many different environments, including your fridge. So what happens when you eat Listeria contaminated food is that Listeria will enter the mammalian host cells, so your gut cells. It can then replicate within the cell, shown here in the cartoon on the bottom left. The blue rods are the Listeria cells. It will secrete different proteins, shown in purple, and then eventually spread from cell to cell and spread throughout the body. And you can see what's really fascinating, the movie on the right, is how Listeria actually moves within the cell. And what's interesting is that Listeria secretes proteins that manipulate the mammalian protein machinery to aid its own survival. So including using these really famous actin tails to spread throughout the cell and then spread from cell to cell. So how does this actually look like in practice? So here's an image that we collected where we infected mammalian cells with Listeria, and we fixed this experiment 24 hours after the infection process. And what we see is mammalian cells shown here in magenta. These are just epithelial cells, and they have been infected with Listeria, shown in cyan. And what's really important here is to recognize the heterogeneity. So clearly some cells have been infected quite dramatically, the cells shown on the bottom, but some of the epithelial cells on the top have not been infected at all. And the question really is, what happened to the cells in the middle? Did they get infected much later and the infection hasn't progressed as much? Or perhaps the cells in the bottom have spread to the cells in the middle, and also that happened more recently. So really looking at these processes live over time can have information that we might find important to understand this process. So that, again, this is like the model system I'm interested in. And with that, I want to talk about a short project on tagging and tracking of secreted proteins that I've worked on in my postdoc. So this is work that I've done with an undergrad student, Dilara Bartan. Here she is at her graduation. She's done really great work doing an honors thesis on this work. And most of what I'm going to show you is data that she collected herself. So we were interested in understanding more about these secreted proteins from Listeria. So these are called effector proteins, and they aid in Listeria maintaining its life cycle. So shown here in a cartoon representation are just different proteins that are secreted, and there are more than 200 proteins that are predicted to be secreted from Listeria. So our question then was, can we label them fluorescently and look at their dynamics? So since I introduced fluorescent proteins earlier, the obvious suggestion could be, okay, why don't we just attach GFP to these secreted proteins to look at where they are? It turns out, though, that this is one of those caveat cases where if we want to see dynamics of the secreted protein, we might affect the secretion process itself, because these effector proteins must pass through relatively narrow secretion apparatus. And by attaching the fully folded GFP molecule, it might get stuck, and we might affect the whole process and dynamics just by trying to look at it. And this is where the split GFP technology comes in. So different labs have shown that when you split GFP up into different pieces, each part will be non-fluorescent, but these parts can come together and a fully complemented fluorescent protein can form. So here I'm showing you structures of GFP. So GFP consists of 11 beta sheets. And if you take off GFP 11, the last strand, GFP 1 through 10 will be non-fluorescent and GFP 11 will be non-fluorescent. But when they come together, they will complement to the fully folded and fluorescent GFP molecule. So now we have a system where if we only attach GFP 11, the last strand, to our favorite effector protein of interest, it is much less likely to be stuck in the secretion apparatus. So we chose a model effector protein called INLC, shown here in cyan, to demonstrate if the system works. The structure has been solved, which was really useful when we wanted to evaluate if attaching the GFP11 strand might affect its folding or function. And you can see it looks pretty non-perturbing. Another reason why we liked INLC as a model is that there's a demonstrated role for Listeria virulence in a mouse model. And also there has been quite contradictory evidence for what its function is in infection, supported by different biochemical assays and studies. So first, there was some data that showed that INLC might interact with cytosolic proteins and interfere with nuclear function. And another study has found that INLC actually binds a membrane-associated protein. There's even a co-crystal structure, and suggested that INLC might perturb cell-to-cell junctions in the infection process as Listeria spreads from cell to cell. So that's why we were interested in using this as a model system. So what Dilara actually did was the following workflow. So we started by transfecting GFP 1 through 10 into the mammalian host cell, which will be the cell that will be infected with Listeria. And again, this is a non-fluorescent majority of GFP. We have then genetically modified Listeria by attaching GFP 11 to the C terminus and used that to infect the epithelial cells. And then we just needed to wait for the infection to progress and to wait to see if the GFP pieces will find each other upon secretion of INLC, and see if we can see where INLC is in the infection, in the infected cell. So
Esther,
yes.
Sorry, quick question from Anne is in the chat here, asking if INLC, if the two functions of it are mutually exclusive.
I don't think so. So the second study has cited the first study and has alluded to the fact that perhaps it does both. My personal hypothesis is perhaps it's dependent on the timing of the infection. Perhaps it does one first and then the other, but it's it is not mutually exclusive. My personal hypothesis, perhaps it does both, depending on context, because the data looks pretty convincing. Each data set looks pretty convincing. So the experiment, the GFP complementation experiment, worked really well. So I'm going to show you just one movie. This was a lot of work that I put into troubleshoot the system, as you can perhaps imagine. So I want to orient you to what we're looking at. This is one epithelial cell with a blue nucleus as a marker. And you can see that the infection has already started. So you can see these really small black rods in the bottom that are Listeria. And this is actually an infection that has a movie that we collected between 6 and 18 hours post-infection. And what we are looking for is, does the infection progress? So does Listeria replicate? And then also, do we see green fluorescence appear over time? And the answer is yes, otherwise I wouldn't be showing it. You can see Listeria really take over the cell, and then you can see accumulation of green fluorescence over time. So we have analyzed some of these, many of these movies, and what we saw is that INLC does go to the nucleus, to the cytosol, but then most importantly, it also goes to the cell protrusions at the edges, if we can see with this white arrow. So really, that's another point of evidence why we think perhaps INLC does both in this context. What's also interesting is that when I introduced GFP, there's more than one fluorescent protein. So another one is mCherry, and people have also designed split mCherry versions, and we were able to show that split mCherry works just as well in this system. And this is work that has been done by Du Mai, who was a really great rotation student. She's a computer scientist who wanted to gain some wet lab experience. So this was a really challenging project for that for that goal, and she succeeded and collected these beautiful images. So with that, we have shown that split FP labeling works well to visualize secreted proteins in infection. It's compatible with green and red fluorescent proteins, and we've shown that the dynamics that we show with that we've seen with INLC are consistent with both biochemical studies, actually, in terms of the localization.
Esther, before you, I'm going to ask a question of my own here. Yeah. So I don't know this about the proteins. Are the fluorescent proteins sufficiently different that you could use two colors within cells, or are they not sufficiently different? And when we end up with like the red and green pieces interchanging?
Depends on the pair. These, I think, red and green could work. We've thought about that, yeah. Like there are different families of fluorescent proteins. You could pair them such that you could choose the 11 for either one as well. We thought about that at one point. Yeah, it depends on which one you choose. Yeah.
And I think Sean has a question here.
Mm-hmm.
Hi, Esther. I just want to make sure. So you didn't see any difference in infection with your fusion construct relative to the, I guess, unmodified or unengineered Listeria. I just want to make sure that the INLC protein isn't, its function isn't perturbed.
Yep, and we tested that, yep.
Okay.
Thanks. So then the main project that I worked on in my postdoc is developing tools to label and track RNAs in live cells. So just like fluorescent proteins, you could imagine that you can gain really important insights in biology by visualizing RNAs in live cells as well. So why is this important? Only a small fraction of the human genome encodes for proteins, and a much larger fraction encodes for RNAs that have functions by themselves. And this really busy cartoon demonstrates a couple of points that I find important, which is that there are many different types of RNAs in cells, and it's important for their function, where they are in the cell, who they interact with, and where they might go in different perturbations in their different life cycles. So really looking at RNAs relative to different markers and looking at where they go over time might give us a lot of insight into RNA function, generally speaking. And this is even something we can think about when we think about the mRNA life cycle. So mRNA is made in the nucleus, and then it has to go to the cytosol for translation, and it interacts with its process and interact with many different proteins and RNAs along the way. So looking at where RNA goes relative to its interaction partners and relative to the localization within the cell can be really important to understand its function. So one example is that in stress, mRNA is actually known to go to these stress granules, which are membranous organelles that are shown here in green. So again, these are these aggregates basically of RNA and proteins that are found in response to different stressors. And even understanding these processes, this will be important later on, but even understanding the formation and dynamics of these granules can be important to understand more about RNAs and cells. And this would be in for the mRNA case. So long-term, the goal is obviously to develop tags to label non-coding RNAs, because those are the ones where much less is known about in terms of its function than coding RNAs. But first, as I developed this tool, I wanted to label and track messenger RNAs, just because we know more about its function already. So like, it's not like with proteins that fluorescent RNAs are known. So actually, I don't think any fluorescent RNAs have been discovered in nature up until now, which means that we have to engineer a fluorescent molecule onto a tagged RNA to label it. So the most commonly used RNA tagging system is called the MS2 system that was developed by the Singer lab. And I'm showing you a cartoon overview of how that works here. So let's say we have an RNA of interest. We can fuse an RNA tag, in this case, to the C terminus to it. And then what in the MS2 system happens is that a second component is produced in the cell, that's a fluorescent protein. And this fluorescent protein is fused to a binding domain, which binds the RNA. And by multiplexing the system, so adding multiple tags of the RNA tag to the RNA, we can now recruit many copies of the fluorescent protein to the RNA of interest to essentially recruit fluorescent proteins on your favorite RNA of interest. And you can imagine that these tags end up being huge. Typically, people use up to 24 copies of the RNA tag, and then they bind dimers of the fluorescent proteins. You get up to 48 fluorescent proteins per RNA, which is great if you're interested in messenger RNA dynamics, because it has been shown that then it's less disruptive. But you could imagine that really this is not a great tool to look at small non-coding RNAs, for example. So this system has been used successfully to look at mRNA dynamics down to the single RNA level. And I'm going to show you one movie that I collected as just an example. So this is beta-actin mRNA that was tagged with the MS2 system, and you can see in the cytosol single spots move around that correspond to single fluorescent spots move around that correspond to single mRNAs, and you can quantify this movement and gain information on the dynamics of these RNAs. I think that was the question. Let me.
No, sorry, Steve Singer is just
Yes,
pointing out that
yes.
this is technically the Robert Singer lab, not the Steve Singer lab.
Correct. Yes. Did I say Steve Singer? I hope I didn't.
No. I think just Singer lab in our department is likely to be interpreted by Steve's labs. Many talents have just expanded.
Okay. So then people have realized that the MS2 system is not sufficient to label and track all RNAs that they might be interested in. So there has been a recent push to develop other RNA tagging systems that are much smaller and perhaps less perturbing for non-coding RNAs. And the prime example are the spinach, broccoli, and mango systems, where a short RNA aptamer is fused to an RNA of interest, just like the MS2 system, but here we only use one or very few copies, and which this then binds a small organic molecule that is non-fluorescent in isolation, but is fluorescent upon binding to the RNA. And this works well for some examples, like you would expect. So I'm showing you here a cell where the 5S RNA has been overproduced, and you can see it being bright green and fluorescent from the broccoli RNA tag. One of the downsides is that it has been developed in vitro, so there's a lot of optimization that's required, and this really only works for RNAs that is produced at really high levels. So we thought there's room for yet another tool, and which is what we are calling Riboglow. So Riboglow is the system I developed, which consists of same as the other techniques, an RNA tag that we are fusing to our favorite RNA of interest, shown here in black. And in our case, the RNA actually binds a quencher, shown here in brown. It's called cobalamin, another name for vitamin B12. And the fluorescent molecule shown here in red is synthetically coupled to cobalamin through an organic linker. And the advantage is that we can change the fluorophore because it doesn't interact with the RNA directly by this design. Cobalamin is actually, the riboglow is actually based on a riboswitch. Riboswitches are these short genetic elements in bacteria where a short RNA tag binds a metabolite to regulate gene expression. In our case, we're using the cobalamin riboswitch. So again, cobalamin is vitamin B12. Here's another illustration of cobalamin in brown. The advantage of the system is that the interaction of cobalamin and the RNA has evolved in nature. So many of the problems that the spinach and broccoli people have seen we won't have, because we know that it folds robustly in different contexts. And riboswitch is actually used in different synthetic biology applications. So they are very portable to be fused to other RNAs in different sensing systems. This was a collaboration with the Beatty lab at CU Boulder, and Rob Beatty's lab had actually crystallized the RNA interacting with cobalamin a number of years ago. So it turns out that cobalamin has fluorescence quenching properties. So the Grissom lab has shown that when they synthetically couple a fluorophore to cobalamin, they see a reduction in fluorescence by about 100-fold. And the degree of quenching is modulated by the organic linker shown here in red. So this is where our hypothesis came from, which is that the cobalamin fluorophore probe will be dim in isolation, but upon binding to the RNA, we hypothesized that we would see an increase in fluorescence because we would stereically separate the cobalamin and the fluorophore and induce dequenching, basically. So this is what we tested. So we started out by generating a suite of cobalamin fluorophore probes. So this was a collaboration with Dorota Gulko's lab at the Polish Academy of Sciences, who is actually an expert in synthetically modifying cobalamin. Turns out those experts exist. She helped us design these probes, and her students synthesized them in her lab for our study. So we modified both the linker, shown here. We increased different lengths and different chemical properties. We changed the chemical properties from PEG linkers to peptide linkers. And then we also systematically varied the fluorophore across the spectrum, basically, to investigate if different trends would emerge. The first thing that we noticed is that all of these probes are quenched in comparison to the free fluorophore. So if you look at, for example, the EDO 590 box, you can see that the red bar is the free EDO 590 dye. And then when EDO 590 is coupled to cobalamin in the gray little bars, they are all quenched. And if we change, and the amount of quenching depends on the length of the linker, as you can see in the EDO 590 example. Like we would expect, right? The longer the linker, the further apart the two can go for, and the less efficient the quenching is. So this is actually consistent with the thread quenching mechanism. If we look at the absorption spectrum of cobalamin, shown here in black, and the fluorescence emission spectrum of the different colors, we can see that there's more spectral overlap for the green probes. So we would expect there to be more efficient thread quenching, which means that the residual fluorescence is much lower, which is exactly what we see. And there's less efficient quenching for the red and far red dyes. Interestingly, the very far red dyes, Psi5 and EDO 633, we also see quenching, even though there's really no fluorescent overlap, no spectral overlap. So there must be another quenching mechanism other than thread, is what we hypothesized. So the next question, yeah.
Oh, sorry, there's a couple of questions here. Rebecca Riggins has dropped one into the chat. First, she says, this is very beautiful. If the Riboglow cobalamin riboswitch has to be appended to the N terminus, could there be transcriptional read-through like there is for PP7?
Well, it is found on the N terminus in bacteria, but our goal was to take it and fuse it to any RNA we're interested in mammalian cells in a 3' tagging system. Does that answer the question? So we want to use it as like an isolated element to fuse it as a reporter element, to fuse it to any RNA in mammalian cells we are interested in. And the beauty is that actually cobalamin riboswitch is not found in mammalian cells, so really we wouldn't mess with its natural function. Was another? Did you have another question?
I'm not sure. Sean had his hand up, but I'm not sure if that's a leftover or a new one.
Okay.
It was left over from before. I'm taking it all in. I'll save questions for later.
Okay, thanks, Esther.
Okay. So then the question is, okay, if we add RNA, what happens to fluorescence? So our hypothesis, like I mentioned, was if these probes are dim and quenched in isolation, if we add the RNA, does it really separate the two, or do we see fluorescence turn on? So the answer is yes. We see increase of fluorescence for these probes upon addition of RNA. And because this is based on a riboswitch, there's more than one RNA sequence actually. Different bacteria have evolved different cobalamin riboswitches. So we varied even the RNA identity and sequence a bit and noticed that we saw fluorescence increase for all of them. There doesn't seem to be an obvious trend, which I think is really interesting for future design applications. So there's no obvious trend in length or even bulkiness of the RNA in terms of the full turn on. So the other thing that we did is biophysically characterize these probes. They bind really tightly to the RNA in the nanomolar range, which is comparable to the MS2 system and the most recently evolved broccoli RNA system. So that's really exciting and important for cellular applications. And then we decided to benchmark the RNA imaging, to benchmark the system against existing mRNA visualization systems. So the question we had was, okay, can we actually visualize mRNAs in live cells, and how does our system compare with other systems? And for this, we wanted to go back to the stretch granule assay for the comparison, but also then assess the question, can we see single RNA, can we visualize single RNA dynamics? So first, let's go back to the stress granules for a minute here. These are the mRNAs that localize to these granules upon stress, upon different stressors. And here's an image of that. So basically these different granules can be identified by marker proteins. So these are membranous organelles. And here I'm showing you stress granules in green. And the green color comes from a marker protein that is labeled with a fluorescent protein. Different granules, in this case, P bodies, are shown in magenta. You can see that they are not overlapping, and they consist of different protein and RNA components. So here we were interested in seeing, okay, can we see localization of mRNA to these stress granules in green? So the answer is yes. So when we label stress granules with this marker protein called G3BP1, we see formation of granules shown here on the left in red. And when we tag our favorite mRNA with Riboglow, in this case 4 copies, we see that it co-localizes to the granule. And we can quantify the co-localization by the fluorescence ratio. We can see that we need at least 4 copies of Riboglow. And we can even look at this process live. You can see formation of these granules here on the left. And this is interesting because like this movie is 90 minutes. And because we're using synthetic dyes, we don't see a lot of bleaching. Again, these are synthetic organic dyes that have really good photophysical properties. So next we wanted to see, okay, how robust is this system? And basically we quantified how efficient the recruitment of the RNA to granules is. For our Riboglow Sci5 system, we saw that the RNA, if there's a stress granule marker, we will see the RNA at that position in all cases. And like I said, because for Riboglow, the fluorescent dye does not directly interact with cobalamin and the RNA, that does not directly interact with the RNA, we can change the fluorescence colors. We've done the same experiment with ENDO590, and we also see that the RNA is recruited to the stress granules efficiently. In contrast, for broccoli, this did not work as well, which is consistent with the literature that broccoli is not very efficient to label messenger RNAs. Most of the granules that we see, we only see the protein marker, but we don't see recruitment of the mRNA by this technique. It does work really well for the MS2 system, though. So in comparison, we think that Riboglow outperforms broccoli, which was exciting at the time. and it performs on par with the MS2 system. So it seems to be a contender for a robust RNA tagging system. So next I wanted to test, can we visualize and track single RNAs in live cells? And for that, I again tagged a messenger RNA. In this case, I attached 12 copies of Riboglow, and then added the cobalamin fluorophore probe. And what we see is, again, we see these nice puncta in the cell that are indicative of single mRNAs. They move around just like the MS2 tagged mRNA would. We can quantify this movement. So these basically these puncta moving around in the cell can be tracked, and we can quantify different features of this movement, so velocity, diffusion coefficient, and those kinds of things. And what we found is that dynamics are comparable to what other people have seen with the established MS2 system when they tag the same RNA, which demonstrates to us that we actually do see single mRNA molecules when we tag it with Riboglow. So next we wanted to see if we can use Riboglow in more complex applications. This was a collaboration with Tim Stasevich's lab at Colorado State, and they have developed this really interesting translation assay where they label mRNA. And so Tim's lab has actually used MS2 to label mRNA. Again, now we're looking at mRNA labeled with an RNA marker. And my question was, okay, can we use Riboglow here? And then when we translate it into a protein, the protein has an N-terminal epitope tag fusion that can be labeled with another color. So as soon as a ribosome makes the protein, you will see the nascent protein being in a separate color. So these translation spots are going to be labeled in 2 colors. And Tim's lab has shown really nice work at looking at translation dynamics. This also turns out this works with Riboglow. So we see the mRNA spots. I'm shown here in red in the top box here, and then it co-localizes with the protein marker in these 2 examples, and they co-move together, demonstrating that we actually do see translation with Riboglow. So with that, the last question that I had was to see, can we actually use Riboglow to look at non-coding RNAs in live cells? So
Esther?
for that, yeah.
Oh, sorry, actually, before you move on, Mark Rose dropped a question in the chat. Can you evolve the ribo switch to have an even better KD to get around the background signal noise problem?
Yes, I think, yes, we could. I think the problem with the background is that the probe is actually not cell permeable. So we use this technique called bead loading. We can talk about that in a minute. If it were cell permeable, we could wash out the unbound probe, I think, which would have the same effect. And for that, you need really tight affinities as well, right?
Mm-hmm.
So,
as long as I interrupted you, Hyen, did you just unmute to ask a question?
Oh, yes. Thank you, Heidi. And Esther, I was just wondering, when the experiment you just showed where both the mRNA and the nascent protein was labeled, I was wondering, how was the nascent protein labeled?
It was an epitope tag that binds fluorescently labeled antibody.
I see. So, but this was presumably in vivo, so it's fluorescent labeled antibody applied in vivo, and well, not in vivo, but live.
Mm-hmm.
I see. Okay, got it. Thank you.
Yeah. Okay. So then I was interested in, okay, does this also work for non-coding RNAs? And for that, I chose USNRNA as a model non-coding RNA. Again, this is a non-coding RNA that's involved in splicing. So again, this is for messenger RNA maturation. This is the step where it's involved in really early on. And it turns out that, again, because splicing happens in the nucleus, there have been reports that splicing factors can also localize to a different type of RNA protein granules in the cytosol. So this is why where my interest came in. And there are many different studies that actually showed in many different systems, in Drosophila, in plant cells, and in different infection conditions, these U bodies have been shown. So in all of these images, we're looking at orange spots in the cytosol that consist of different spliceosomal RNA and protein components. And these studies have been done with fixed cells because no tools are available to tag non-coding uRNAs. And this is why I thought this could be a good application to test Riboglow. So then the long-term question could be, what are roles of these U-bodies? Are they involved in quality control? Do they perhaps regulate splicing? But first, can we use Riboglow to label and visualize these granules as well? So for that, I've used U1 as an RNA with Riboglow, and then tested if we see cytosolic granules. And the answer is yes. So in the untransfected control, we see background fluorescence from the unbound probe. But then as soon as we add Riboglow tag with U1 to the system, we see these cytosolic spots shown here in red. And to convince myself that these actually are U-bodies and not just random aggregates, I co-transfected a marker protein called SMN, which has been shown to localize to these granules, to these U-bodies with GFP, and we can see co-localization of the color at the granules. So to me, this means like, oh yes, Riboglow is compatible with tagging and imaging non-coding RNAs in live cells. And so basically, Riboglow is a new platform that's modular to visualize RNA in live cells. Interestingly, for future engineering applications, all of the components are modular, right? You can modulate, like someone asked earlier, you can modulate how RNA and cobalamin interacts without changing the fluorophore. You can change the fluorophore without messing with the RNA cobalamin interaction, etc. So they can be optimized separately. And I was able to show that we can tag and visualize mRNA through its life cycle. And this works on the single mRNA level as well. And this was, at the time we published it, was the first study to fluorescently label US snRNAs in live cells. So then what I'm interested in working on in my lab now is to look at dynamics of coding and non-coding RNAs using Listeria as a cellular perturbation system. So it turns out that, like I mentioned, these U-bodies have been shown in response to Listeria infection, for example. So that could be interesting to look at. And because US and RNAs are involved in splicing, there have actually been reports that showed that splicing changes in response to infection as well. So I'm wondering if that is connected. And generally speaking, tracking coding and non-coding RNAs in these complex systems using this Listeria model system. So with that, I want to first thank my postdoctoral lab and my collaborators back at CU Boulder, because that's where I've done this work. So this is Amy Palmer is my postdoctoral advisor. This is her lab. My collaborators, Rob Beatty and his students, especially Jake Polaski. And then Rota Gruko was the organic chemist whose student has made the compounds. The Listeria work was done by Dilara Batan, my great undergrad student who I worked with really closely, and a number of collaborators back in Colorado. And then now I'm working with my own lab, which is really exciting. We haven't actually started anything. I'm still working on getting access to the lab. But here are students that I'm working with remotely right now. Nadia Safras is my first grad student. And then there are a number of undergrads who I'm working with as well. So with that, I'm happy to answer more questions that you might have. How am I doing with time? Okay.
Yeah, your timing was perfect here, Esther. So thank you so much, first of all. The biology department usually has all the little reaction buttons going on. But let me see. Oh, Jeff Long is both clapping and raising his hand. He has 3 hands at the moment. Go for it, Jeff.
Hi, Esther. That was a really nice talk.
Thank you.
You mentioned that you're unable to transfect it, right? You have to sort of load them. Can you explain how that works into your
Yes,
cells?
let me see if I kept that on here. Yes, here it is. So this is actually a technique that I learned from Tim, who does this with fluorescently labeled antibodies. And that's what got me the idea. So it's basically, it sounds high tech, but it's not. So it's basically your imaging dish, and then you have this like very low tech, what's called a bead loader. It's basically an imaging dish with it's like a salt shaker where we have these microbeads that you tap onto the cells, And then like you literally load the cells like this. You generate these really small ruptures in the cell in this way, and then any fluorescent molecules will enter the cells in this way. And then you add fluorescent media, add cell culture media right away to heal the cells, and then rinse, and it works really well.
But you can't. Have you thought about just electroporation? Is it possible
Yeah.
to do that?
I have thought about that and tried it. The problem was with these epithelial cells, by the time they have settled down and attached to the surface, I was afraid that the dye had gone to like weird places, which is why this seemed very acute and I could go and image right away, basically. And it worked so well that I kind of just stayed with it.
Okay. Yeah, I mean, it's very cool. It'd be really great if you could transfect it or electroporate it.
Yeah.
And then the second thing, just quickly following up on, you're looking at uRNAs. Have you looked at use the system for RNA transport? For example, you know, like in Drosophila embryo, you have bicoid and nanos that go to the anterior versus posterior part of an animal, or in oligodendrocytes, in our case, the RNA actually traffic on the microtubules. It'd be kind of cool to apply this technology to look at real-time transport of RNA on microtubules.
Yes, that sounds exciting. Yes, that sounds really interesting. I haven't thought about that, but yes.
We could chat.
Yes. Can you beat load yourself? Is the first question.
Well, let's try to improve the technology.
Yes.
Okay, thank you.
You've definitely captured the attention of our neurobiologist, Esther. Hi-En, and then Tom. I see your hand too.
Hi. So, Esther, I'm not sure I completely got how exactly the technique works. So you do have to synthesize the mRNA the target mRNA you're working with the linker, right? So that it'll interact with the fluorophore. Is that right?
I'm gonna find the cartoon that explains it. So it's are you familiar with MS2 or RNA tagging in general? Okay.
Really?
So it's basically, you have to genetically modify the RNA and attach an RNA tag genetically in this way. So the black part is an RNA tag that is genetically fused to the RNA of interest. And then this component is added to the cells.
All right, I missed the genetic part. That's actually the part that I was most interested in, because then it has the potential capability to be used in vivo. Right?
Yes. And like, my idea is there's more than one riboswitch, right? There are these riboswitches that bind all sorts of metabolites. That could be an obvious way to change it and optimize it to make it cell permeable, for example.
Okay, so the other question I had was that you mentioned that this modification will be preferably happen on the 3' end, right? Is that?
It could. I mean, there's no, I think from the RNA tag perspective, there's no preference. The U1 actually I put on the 5' because I've seen someone else put a pull down RNA tag on U1 before, so that's why I used the same position. There's no preference from the tag perspective.
I was just wondering, like either the 5' or 3' end of the natural mRNAs, they often carry with them the regular sequence. So have you guys observed any like changes in terms of stability or regulation on those mRNAs with the modification?
As far as I know, I haven't. But I tagged RNAs that have been tagged before in similar positions. So like for the mRNA, people have tagged in the 3' UTR before. That's why I used the same position. I even used similar RNA. Like Ben-actin mRNA has been tagged with many different tags at that position before. And like I said, same, yeah.
Oh, sorry. Last question. If you after you load the fluorophore, how long does they last in the cells? I'm actually thinking about in vivo system here because we have in vivo live imaging system. I know you probably haven't tried it in vivo.
I have not. I think there's no. I think it will eventually go to the lysosome, and you have like these off-target fluorescent lysosome labels. Eventually, I think I would expect, or like you get cell division and then it'll dilute out. But they are like fluorescent synthetic organic dyes.
When they're not binding. on the fluorophore themselves supposed to be quenched. So even if they end up in the lysosome, presumably they're still mostly quenched. No.
Well, it kind of depends on which one. So they are quenched, but not like dark. So you can see here, for example, the ADO591 is quenched. That's the one I've done the most work with, but it's only about 10% fluorescent of the synthetic dye, and 10% of like a really bright organic dye is still quite a bit. And like the Sci5 one is quenched, but not down to 0. It depends on which one you choose. And then depending on which one you choose, the full turn on is also different. Yeah.
This is fascinating. We got to catch up later too. I'll yield.
Okay, Tom, you got to take the microphone away from Hyena. We're gonna.
Oh, yeah.
we have a new collaboration, I think, starting up here.
Thanks, Esther. This is really fascinating. I was fixated on the interaction of the CBL domain with what from your cartoons look like the RNA secondary structure, and it made me wonder how, yeah, if you find one of those cartoons of the secondary structure, oh yeah, that one's good.
Yeah. Okay.
You know, it made me wonder to what extent, given the quenching abilities of the CBL domain, how much, if the secondary structure fluctuates, if it does at all, would that lead to quenching? And then would you have a scenario where you have binding, but then no fluorescence? And, you know, maybe there would be some disadvantages to that, but also maybe some advantages where you could track secondary structure. I actually don't know how much secondary structure fluctuates.
So that's something we haven't explored. So this basically means what is the mechanism of quenching, right? That's the question. And how does binding RNA affect quenching?
Well, no, I was imagining that you might have binding but no fluorescence if binding depends on the structural stability of the RNA secondary structure. Meaning like the riboglow could be there, but quenched because the RNA is tweaked.
Yes.
And maybe that would be bad, and maybe that would be interesting.
Yes. We haven't done that. It could be interesting. Yes, could be interesting. We've actually thought, like right before COVID hit and I ended up moving and all of that, we had an idea to modify cobalamin at different positions outside of the 5' hydroxyl down here where the fluorophore goes, and see if we can make 2 different versions that bind and then evolve 2 different RNAs and then do multicolor. Does that kind of get at you? Yeah, basically mess with the cobalamin structure a little bit more. Yeah, that was, yep.
Well, maybe. I think actually if you could do it, tweak it so it bound the same RNA but in a different spot.
Oh, yeah.
You might get to see a little bit about how RNA structure affects your detection.
Mm-hmm. Yeah, yeah. Yep, that could be. Yep.
Anyway, so glad you're in the department. Welcome.
Thanks.
Okay, Mark.
Hi, Esther. Beautiful talk, and welcome to Regents and Georgetown. So I guess I'm a little hung up thinking about the problem of this impermeant dye, which I feel like is going to really be a barrier to its adoption in a lot of systems. So I guess I have a 2-part question. So one is, have you thought about trying to make it permeable? Probably yes. But the second part is, then I was thinking about maybe there's a more direct route, which is that you're basing this on relief of quenching, but there are other systems, and we'll talk about this a little bit tomorrow, when we're using, where the fluorescence arises because the fluorophore binds, and it's now in a different environment than it is in the aqueous environment, and now you get this big boost of fluorescence. So that's based on being in a hydrophobic environment within a protein. But now I really don't know what happens when a fluorophore is bound to a riboswitch. But if that were true, then you could get there directly without have a much smaller, perhaps permeant fluorophore.
Yes, that's a good idea. Yeah, it'd be interesting to hear you. So one thing we thought about in terms of making it permeable is to use like cells take up cobalamin through pathways to use that. But we kind of stopped thinking about that when we realized maybe all we'll do is label the cobalamin uptake pathway and get all sorts of like weird artifacts. So that's sort of why we didn't pursue that further. But yes, I've thought about how to make it permeable a bit. Yeah, or like the peptide linkers between cobalamin and the fluorophore could be cell penetrating peptides, those kind of things, right? Yeah, we thought about that. But yes, your idea about thinking about the quenching mechanism is interesting too.
Esther, I feel like the question I've got has probably been, you've addressed it kind of in part through your other questions, but how sensitive is this to RNA decay? Because is this a tool, because I realize you're looking at localization. How useful is this to kind of track RNA decay rates or differential RNA decay rates?
I haven't tested that directly. I think we need to. I know that for the MS2 system, the affinity between the probe and the RNA matters because the MS2 folks have tested that extensively and show that if it binds too tight, then it'll stay there and won't get degraded. The tag won't get degraded. And now you're looking at RNA pieces basically doing the degradation pathway. So yes, I need to test that with Riboglow, and affinity matters in that case.
Are there other questions? I'm like looking in here, and I know we always allow a little bit of breathing room, but we try to get done by 1:30. And I think, Esther, you may have done like the semester's best job of bringing it in on time here. Exactly. Are there? The
students will love her.
I was going to say, so folks, so Zach, did you send around, did the grad students know where to go for the Zoom hour?
Yes. Yeah.
Thank you, Zach. Okay. So grad students, now you know both about the cool research, and also Esther's just great to talk to about science and creativity and career paths and things like that. And she's just gotten here. So I think it's a great opportunity. Esther, I'm so glad that you joined us today. This is great. One of the things we try to do is get to know our new colleagues well. And I think today you've hooked the bio department on the latest chemistry hire. So thank you so much for joining us. To the rest of the bio department, thanks folks for coming today. Hope you make it through the next day and a half, and spring break lies ahead. So thank you, everybody. Thank you, Esther.
Thanks.
Thanks, Esther.
I just wanted to say hi to Esther. Esther, I'm Martha, the former resident of 532, and I hope that you're getting settled and enjoying the sunset view out the window, and that you're working on the 5th floor.
Thank you.
Did you leave her a little gift or a little letter in the desk the way
exactly
residents do?
I copied the one that Trump left for Biden.
For Obama. For Biden.
No, I did. I decided not to follow that tradition, but I did leave her some millet seeds on the carpet floor.
And you might be getting some cicada cookies, Esther.
That would be, I will bring you chocolate covered cicadas come May as a little welcome.
Thank you.
This is actually, Esther, a thing the biology department does. We eat chocolate covered cicadas once every 17 years. So
it's a long-standing tradition.
As an honorary member now, we'll welcome you into the club.
So Esther, we also have a microbial interest group.
Oh, yeah.
And it strikes me that your interest in Listeria, that would really, fit with our meetings, and so we should talk about trying to incorporate your group in that.
Okay, thank you. Yeah,
I agree. We have, and you and I will share some, I think, intersecting interests. And I'd be interested. I'll talk to you later at 3:30 about how we might use this to look at direct targets for small RNA in Staph aureus.
yeah, That sounds great.
Sure. I'll see you at 3:30.
See you then.
Thank you, folks. Have a good day.
Thank you.